interested in is how to start an immune response, and more specifically, how to start an immune response and kill cancer cells. So what we see here is the final stage of this process. So in red, we have a target cell, and in green, we have cytotoxic T cells that have been activated. They go around finding their targets and killing them. So this red blink that you see and the red cell disintegrating, this is indicating cell death. So these cytotoxic T cells are very potent, and they can go through multiple rounds of killing multiple cells. So you want this process to be very tightly controlled. The way that cytotoxic T cells recognize their target cells is through this interaction. So every cell on the surface expresses this protein called MHC class I. MHC class I binds and displays short peptides that are coming from different proteins that are expressed in the cell. So this short peptide could be coming from a virus, or coming from bacteria, or in case of cancer, it could be coming from a mutated protein that will be only present in a cancer cell. So now T cell has a protein called T cell receptor, TCR, and only when this TCR binds to MHC, displaying a peptide that the specific T cell recognizes, only then it can go and kill this specific cell. So in a teaspoon of blood, we have about 3 million of T cells, and they all have different TCRs. And you don't want these T cells to be activated accidentally, because this would lead to a muscle tissue death and basically person death as well. So these T cells exist in so-called naive state. A naive T cell is not very metabolically active. It doesn't express all these molecules that are required to kill its target, and it just surveys the body ready to start to attack a potential invader. The way these T cells are activated is by a different type of immune cell called dendritic cell. So dendritic cells have to express the same MHC class I peptide combination in order to find the right T cell, and these T cells then proliferate, and then they can kill the target cells. So one important step of this process is for dendritic cells to take up fragments of cells that are potentially infected or coming from a tumor. So through this cycle, dendritic cells are very important orchestrators of immune responses. And pretty much all the tissues in the body are full of dendritic cells that survey the environment for the signs of danger. They sample both healthy and unhealthy cells, and when they sense pathogens or different danger signals that could be coming from tissue damage, then they migrate into the lymph nodes. So in proximity of all tissues, you have loads of lymph nodes. And then in the lymph node, they will process these fragments of cells that they've taken up, present them with their MHC class I molecules. And in lymph nodes, they can encounter thousands and thousands of T cells in order to find the right T cell with the right T cell receptor. These T cells start proliferating massively, and this is what leads to lymph node swelling during infection or cancer. So such activated T cells then exit the lymph nodes, enter the blood vessels, and then they can infiltrate inflamed tissues and kill the target cells. We have recently identified many diseases where, many patients which are deficient for dendritic cells. Also in mice, when you deplete dendritic cells, this leads to susceptibility to cancer, infections, and also autoimmune disorders. So this really highlights this critical role of dendritic cells in initiation of immune responses. So this is what we are interested in. We would like to understand how these different steps of dendritic cell function contribute to initiation of the immune responses. So what I described is a process that's efficient, right? But many viruses will actually manipulate this pathway in order to evade the immune response. And also, it doesn't necessarily work very well in case of cancer. So what we hope to understand is the molecular details of these processes, so we can manipulate them and also try to improve the efficiency of, for example, vaccines or therapeutic strategies. So one step that we think is particularly important is this process of taking antigens from cells and presenting them on MHC class I in dendritic cells. So how does it work? So all cells, as I mentioned, they will present MHC with peptides on their surface, right? So here the goal is to just provide a sample of different proteins that are present in the cell, and then by chance, if you have a virus, you will also have fragments of the virus presented, or these mutated proteins, as I mentioned. So these peptide fragments come from degradation of old proteins or newly made proteins that were accidentally didn't fold properly, and they will be degraded by a machine called proteasome, and short peptides will be loaded onto MHC class 1, and then they will go to the cell surface. So during viral infection, if a dendritic cell is infected with a virus, the same pathway can be used in order to activate the T cells. But then during cancer, dendritic cell doesn't actually express the mutated proteins, right? So then it's very important that it takes these proteins from the surrounding tissues. And this is a step that has been observed about 40 years ago, and we still don't understand very well how this happens. So it does seem that these proteins are also processed by the proteasome. But what's surprising is that they have to somehow exit this compartment. So when they're taken up, they go into a vacuole, and normally they would be degraded there. And the reason why it's so important is that you have a lot of toxins and viruses present in the fluid that you don't necessarily want to allow them to enter the cytosol, right? So generally, these membranes they function as protective barriers. But in dendritic cells, they seem to somehow allow proteins to enter the cytosol so that you can present these peptides and activate T cells. So we thought that this process of antigen entry into the cells would be what's inefficient during initiation of immune responses against cancer. So we looked for drugs that would actually enhance antigen entry, and then we looked whether that would lead to a more efficient anti-cancer immune response. So we screened the library of FDA-approved drugs, about 800 drugs, and we identified about 30 compounds that increased antigen entry into dendritic cell cytosol. What was quite surprising that among the drugs we found things like Prozac and also a lot of other antidepressants and antipsychotic drugs, which are generally known to have a lot of off-target effects. So we focused on these compounds from quinazolinamine family because they were our most potent enhancers of import. And here what you can see, so here we have dendritic cells in green, and this green protein that they're expressing will mark compartments that became damaged. So we are looking whether indeed these drugs are making endosomes leaky. And so we treat the cells with prazosin, and you can see that in all the cells over time, we see appearance of these damaged vesicles that light up in green. So this confirms that our drugs indeed promote leakiness of the compartments. And now the next question was whether that leads to more efficient T cell response. So we treat our dendritic cells with the drugs, and we give them ovalbumin, and then we culture these dendritic cells in the presence of T cells. So the question we're asking now is, does the drug increase the efficiency of T cell proliferation? Right? If we are going to present more MHC class 1 peptides, we would expect that the T cells will proliferate more rapidly. So here we're looking at T cell counts, and in the absence of drugs, we see some T cell proliferation with increasing amount of antigen, and this is dramatically improved with prazosin. So the next question we asked is whether we can actually use prazosin to improve anti-tumor immune responses in vivo. So the idea is that by enhancing antigen import and enhancing T cell proliferation, we will have a better control of tumor growth. So in these experiments, we injected tumors subcutaneously in mice, and then we followed tumor growth. So here you can see that in control mice, you see it's not very bright, but you can see that over the period of about a month, the tumors are growing rapidly. We then took these mice and treated them with prazosin when the tumors appeared. And in 6 out of 9 mice, we see actually control of the tumor growth, which we did not see in no-treated mice. So this was very exciting, and we wanted to see whether this would also work for tumors that are quite resistant to the immune systems. So one of the reasons why the immune system can't fight off the tumors is because many tumors will actually express molecules that inhibit the T cells. So here's an example of such molecule. It's called PD-L1. It's a so-called checkpoint molecule. And when the tumor expresses PD-L1, it interacts with a molecule on T cells called PD-1. And that prevents this T cell from killing the target cell. So recently, there has been this great success of immunotherapies. And what these immunotherapies are doing, they're actually blocking these interactions. So anti-PD-1 antibodies, they prevent the interaction between PD-1 and PD-L1, and they allow now T cells to kill the cancer cells. So and as I mentioned, these immunotherapies now have provided long-term remissions in many patients. So one of the main differences between immunotherapy and a classical therapy is that classical therapies, they sort of work in the majority of patients, but they increase the lifespan only by a few months. With immunotherapy, in many patients, you see long-lasting remissions, but the problem is that it only works in a small proportion of the patients. So what recently is becoming more and more exciting is that by combining a classical therapy where you suddenly trigger death of a lot of cells and immunotherapy, you might actually increase the proportion of patients in which it works. So we wanted to see whether we could combine this anti-PD-1 therapy where we prevent this T cell, this block of T cells, with our drugs that enhance antigen import and improve T cell proliferation. So here we chose a cancer line that's a bit more aggressive. So it's a B16 melanoma. We again injected subcutaneously in mice. And we started treating them with either prazosin, either or PD-1, or both drugs simultaneously. And what you can see here that for B16 melanoma, neither the checkpoint immunotherapy, anti-PD-1, nor prazosin alone don't actually affect the tumor growth. But when we combine the two, we see inhibition of tumor growth in the period of the 4 weeks. So this was quite exciting. And it suggested that this mechanism, that this step of antigen processing and presentation is indeed quite important for initiation of anti-tumor immune responses. And it suggested that if we sort of artificially facilitate antigen import into the cytosol, we can make T cells proliferate more rapidly. But what we'd like to know is how does it actually happen physiologically, right? So do the membranes also become somehow destabilized and leaky, but in a controlled way? Or maybe there is a channel that allows these proteins that have been taken up to enter the cytosol in a more controlled way. So this question has been very difficult to address because dendritic cells are very difficult to work with. So it was difficult. It's difficult 3 times in one sentence, but that's because it was very difficult. So they are difficult to work with because they die very easily. But with the recent development of CRISPR-Cas9 gene targeting strategies, we can now do gene editing experiment also in dendritic cells. So we needed an assay. So our goal was to set up a screen where we could look for genes that are important for the step of antigen entry from endosomes into the cytosol. So we needed an assay to monitor whether a protein entered the cytosol. And this is not a very efficient process, right? So we couldn't really use microscopy to see them because only very few molecules will actually make it into the cytosol. So we took advantage of a very potent toxin made by plants, which is called supportin. So supportin, it doesn't have a mechanism to permeate membranes, but when it enters the cytosol, it inhibits ribosomes. So it inhibits protein translation. The cells can no longer make proteins, and ultimately they will die. And you need only a couple of molecules of supportin, maybe even one, in order to inhibit all ribosomes and arrest the translation. So we decided to use supportin in our screen. So we generated a library of dendritic cells where each dendritic cell had a different gene edited. So in order to introduce mutations and prevent expression of that gene. We then selected the cells that were resistant to supportin, right? So where supportin could not enter the cytosol and inhibit translation, and cells that were sensitive to supportin. we looked at which genes are being edited in those two populations and which are enriched in cells that are not allowing supportin to enter the cytosol. And we found one gene, a protein called MPEG-1. So MPEG-1, in fact, very excitingly for us, turned out to be a pore-forming protein. So what you can see here is an oligomer of 16 MPEG-1 chains. And this is a membrane. This is the MPEG-1 ring. And it can actually form a pore that will allow transit of proteins across the membrane. And here, to scale, we have our supportin. So it would actually fit the lumen of the pore. And here we are looking at the pore from the bottom, so from this side. So this was very exciting because this suggested that maybe MPEG-1 is a channel that is expressed in dendritic cells that allows proteins to enter the cytosol and provides this mechanism in which this process could be regulated. So indeed, MPEG-1 is actually very highly expressed in antigen-presenting cells, such as dendritic cells, and not in other cells of the body. I'm just going to show you one experiment that we did in order to show that MPEG-1 might indeed be involved in this step of protein transport. So here we're looking at supportin-induced cell death, right? So on the y-axis, we have % of live cells. On the x-axis, we have concentration of supportin. With the increase in concentration, after 24 hours, the wild-type cells die, whereas the MPEG-1 knockout cells are protected, right? So the supportin is not getting in as efficiently. Here we're using a different toxin that also will lead to cell death once it's in the cytosol. And again, the MPEG-1 knockout cells are protected. So we also used a membrane-permeable molecule that could arrest translation, but it doesn't, but it can enter the cytosol spontaneously. And here, the MPEG knockout cells are actually not protected from cell death. So we now proposed that the mechanism by which dendritic cells are actually taking up and processing these antigens for presentation involves a pore-forming protein called MPEG-1. And this might not be very exciting for you, but it has been very exciting for us because there has been really loads of mechanisms proposed, and none of them have gained wide acceptance in the field. So we are now trying to understand in more detail how is actually MPEG activated, right? So you don't want to have a pore-forming protein crazily making pores in the cell. So there must be a very strict mechanism by which you control MPEG-1 activity. Is it differentially regulated during infection and during cancer? These are the questions we're trying to address now. And more generally, we would like to understand other steps that might be not very efficient during initiation of anti-tumor immune responses. So we know that cancer cells actually can modulate dendritic cell functions and, for example, inhibit antigen uptake, inhibit antigen processing. They can also secrete cytokines that will change the way in which dendritic cells differentiate, that will, for example, prevent them from migration into lymph nodes. So our goal is to understand those different steps in molecular detail so that we can design better therapies and also both against infections and tumors. So with this, I would like to thank all the members of my lab and also my previous supervisors, and thank you very much for your attention. Take questions.